Okay, good afternoon, ladies and gentlemen, viewers at home. This is another opportunity to get ourselves once again, where we will be discussing the challenges of answering examination on immunology and public health. Today, I'm going to divide my focus. I'm going to divide it into seven major sections. And this time around, the first one we are going to look at, this is a examination focus. Okay, examination focus on the immunology and public health. And just like I said, we are going to have 7 different sections that today's focus is going to be on. The first one is we are going to look at what are the components and mechanisms of the complement system under which we are going to focus on the relationship between complement system and other arms of innate immune system, other arms of immune system. Knowing fully that immune system we are referring to here is innate as well as adaptive. So we are going to look at that. After that, we are going to also look at likely area of focus on the biochemistry, the genetics, as well as the immunological basis of ABO blood grouping, as well as the rhesus blood grouping system. So then we will also look at we should also focus on autoimmune diseases. So you should focus on that as well. After that, I want you to also expend energy on the classes and functions, classification and functions of immunoglobulin, the 5 immunoglobulin. I want you to focus on what are those classes and how do they take part in the immunological reaction. Then the fifth part is going to be centered on adaptive stroke innate immune system. So I'm going to also give you what to focus on that. Then you should be able to look at the application of immunology, in laboratory techniques, lab techniques. So I'm going to tell you the area you should definitely focus on. Then the last aspect, which is going to be so the seventh aspect is going to be immunization and vaccination. That's one is this one is we're already entering into public health. Okay, so after that, the roles played by Mary Mylon in the transmission and of typhoid fever. That is in the area of public health. Okay. Then the last aspect that I also want you to focus is you should be familiar with basic terms and terminologies. Basic terms and terminologies in public health. So having these 9 broad sections, the first one is complement, the mechanism and component of a complement system. So what are the relationship between complement system and the other arms of government, that is between innate immune system as well as the adaptive immune system. The second focus should be on the biochemical, genetical and immunological basis of ABO blood grouping as well as resource blood grouping system. Then you should be able to focus on autoimmune diseases. What are they? What are the case studies? Give us examples. What are the clinical manifestation of each of these? Then the next one is the classes and functions of immunoglobulins. What are the classifications and function of immunoglobulin? Then you should be able to focus very well on adaptive and innate immune system. So we'll now look at the application of immunology in laboratory techniques. Okay, then we'll also try and see the relationship between immunization as well as vaccination. What are the basic principles of immunization as well as immunization? Then under public health, there's going to be a focus on typhoid fever. So what are the roles played by Mary Mylon in I think 17th century, Mary Mylon in the transmission of typhoid fever. Then we'll also look at the basic terms and terminologies of in public health. Basic terms and terminologies in public health. So all these areas is what I want you to actually focus on. I think you are going to have 2 sections, A and B. Okay, A and B. 1, 2, 3. So out of all these will be synchronized into 6. So it's going to be synchronized into 6, out of which you might be asked to choose from section B. You could be asked to choose 2 from section B, and basically to answer all in section A. It depends, but I want you to know that strictly this is exam focused, and that is why you should just try as much as possible to get all your materials ready to actually focus on these 9 areas. So just like I said, these 9 areas will be synchronized into 6. Then in terms of grading system, our grading system, you are going to have quiz stroke what is called CBT. That one is going to be, I think, 40 MCQ, that is multiple choice questions, 40. That will be later graded as 20 marks. Okay. Then the next one is going to be the attendance, which is going to be 10 marks. Then the other aspect has to do with the basic task, like the assignments, you have been given. That's going to be 10. And the whole of the exam is going to be 60 marks. It's going to be 60 marks. So that is how the grading system is going to be. Now, let's now try as much as possible to make a kind of synopsis. That is, I just want to make a kind of emphasis. It might not be a detailed emphasis on all the 9 topics, broad topics or area of focus that is going to be synchronized into 6, so that you will be able to know, you'll be able to be rightly guided, that you don't need too much notes, you don't need to write history on anything, you don't need to write history or write too much on it. So I want to believe that you are going to focus on, so the first one, which is going to be components and mechanisms of the complement system. So if you have any question on this, how should you attempt it? Now, if you have any question like this, the first thing is that you should be able to list the components. These are two keywords. You should be able to list the component first, then their mechanism. If you want to know mechanism, that is, how do they work? How do all these components, how do they interact with each other in order to be able to carry out its function as a complement system? Now, just like I told you earlier on, that the components of complement system are like 30 different proteins. 30 different proteins, and all these 30 different proteins, they have between C1 and C9. I want you to read more on this. C1 and C9 proteins. All these are very, very possible because they exist in cascades. And that is telling you that immediately one is activated. You just discover that each of these is going to activate others until it gets to the last protein. Primarily, C3 play a very vital role, plays central role in the complement system, because it is this one that is further differentiated into C3A, C3B, and C3C. By the time you read more on the complement system, you'll be able to talk about that. So then, after the components, the next thing I'm expecting you to focus on is the activation pathway. Activation pathway. How do they get activated? So, and don't forget there are three classical pathways of complement system. The first one is called classical pathway. The second one is called alternative. That is, if not the classical, what is the other alternative that this kind of very useful pathway or complement system, what do they which other way can that be? Then the last one is lectin pathway. Okay, so already, if there is any question on this, you're already answering it. The first one is to look at the components. The second part of it is to also talk about the activation pathway. How is a complement system, how is it being activated? Then the C aspect, you should be able to talk about the role of complement system in the immune defense or in immune system. So you should be able to tell us the role played by complement system in immune system. So don't forget, you have to focus very well on what is called opsonization. Okay, this has been variously described for you in details in most of the videos. Okay, then you should be able to look at the role played by complement system in inflammation. What are the roles that they play by that? And basically, after that, the fourth aspect that you also need to focus if you want to answer any question that has to do with component and mechanism of the complement system is that you have to focus on the regulatory mechanism. How is this being regulated? So don't forget that you're supposed to have known why processes such as this must be regulated. So what are the mechanisms of regulatory pathways, mechanisms? Okay, and this one has to do with C1 inhibitors. Then you have to look at what is called DAF. Okay, which is another way of regulating complement system. The first one is through C1 inhibitors. The second one is DAF, which is DAF is a short form of decay accelerating, and F is for factors. So that is, what do we mean by that? That is decay accelerating factors. So that is how to solve if there is any question on the components and the complement system, component and mechanism of complement system, the component and mechanism. So if there is any question on complement system, that should be the area of focus. The area of focus should be into 4. The first one is you look at the components, and I told you that the components of a complement system are 30 different proteins which are intricately working together in form of a kind of network. So, and these complements you have is being numbered from C1 to C9, okay, and it occurs in cascades. And you should know that C3 will play a very pivotal role, plays central role in the complement system. And I said that is the reason why it is being divided into three. We have C3A, C3B, and C3C. Then, after looking at the components, you have to talk about the activation pathways. And there are three activation pathways. The first one is a classical pathway. Then the second one is an alternative pathway, as well as the last one, which has to do with the lectin pathway. You need to know that. Then you also need to emphasize on the roles of a complement system in immune protection. So, and when you are looking at that, you should not forget to mention what is called opsonization. How does opsonization take place? What are the main essence of opsonization? Then you also need to talk about inflammatory or inflammation, role played by inflammation. And under this inflammation, you should be able to mention some of the specific proteins that are involved in the opsonization and the inflammation process. Okay. The fourth aspect is that you also need to look at how is the complement system being regulated, just like you have it in the microbial genesis, and which I told you that immediately after any gene is being expressed, the next thing is to make sure that the system is brought to the normal after complement system, after cascading of a complement system in trying to assist the innate as well as the adaptive immune system. So how can it be regulated? And I said it has to be regulated by the use of two basic mechanisms. The first mechanism is through C1 inhibitors or inhibitory process, then followed by decay accelerating factor. So that is that. If you have any question on the complement system, I'm very sure you cannot run away from having compulsory questions on this, because it takes a kind of special, because of the special role it performs in as a kind of midpoint between innate and adaptive immune system. Okay, so let's go to the second area, the second section we need to look at. Because of our time, I'm going to be a bit faster this time around. Okay, now the next one after that should be the biochemistry of the immunology, as well as the genetic basis of ABO blood grouping, as well as resource factors. Okay, now you need to know that there are two different things that you are talking about. We want to look at what are the genetic basis. Three things, three aspects. You look at the genetics, you look at the immunology perspective, then you should also look at it from the angle of biochemistry. Okay, so the same thing with resource factors. So when we look at genetics, that is, on what chromosome is it being found? The gene that code for ABO blood grouping, what chromosome is it being found? On what chromosome is it being found? As well as other. Then what are the basis, the hereditary basis of it? Can this ABO blood grouping character or traits or inheritance, be passed from their parents to offspring. That is talking about the genetics. Then the immunological aspect of it, that is when you will be looking at antigen antibody reaction. Somebody that is A, what antigen does it have? Somebody that is B, what antigen does it have? Somebody that is O, what antigen is it lacking? Then you also look at it from the angle of antibody. Somebody that is group A, which antibody is it having? Somebody that is of group B, which antibody is it the person is going to have on their serum or plasma? Then the third one is somebody with O. What's that? Then biochemistry. Biochemistry is what is going to let us know the types of sugar or carbohydrates that we have on it. Somebody who is blood group A, what sugar is the person having? Somebody with blood group AB, blood group B, blood group O, what is the biochemistry? That means you need to make some drawings here, sketches. Don't let me say drawing. You need to make some sketches, very, very important sketches. By now, you should know that when you have RBC, somebody is A, so it's going to have different types of sugar in different arrangements. So you should be able to show that. And when we now talk about rhesus blood group, okay, the rhesus blood group is such that biochemically or biochemistry is that the person is having an antigen that is called antigen D, which is standing for rhesus factor, which is antigen D. And somebody who's having that, what kind of antigen antibody reaction should you be expecting that person to have? Please, I want you to go back to the specific video that has to do with the biochemistry of rhesus factors, blood grouping factors. I told you that in that video that we have more than 100 different groupings. So you should be able to actually tell us more about that. So just like I said, that we are not going to waste time because of our time. So let's quickly go to what is expected in section 3, and that is what is going to be on autoimmune diseases. Autoimmune diseases. Okay, so from the word auto means self. Auto means what? Self. Okay, this is when there is a problem. That is when there is a problem for the immune system to be able to differentiate self from non-self. So it is going to result into different types of immunological abnormalities, and that is nothing but something that is regarded as innate, that is regarded as autoimmune diseases. Okay, and when we look at this, that one is going to, so what we want to do is that under that you should be able to tell us, give us the definition. What happens? What is autoimmune diseases? You should focus on the definition. Then you should also tell us the pathogenesis. When you say pathogenesis, patho is pathology, that is from diseases. So that is how the genesis of this kind of autoimmune diseases, you should be able to tell us, including their clinical manifestation, how does it actually happen, and all this. You should be able to tell us definition, the pathogenesis. Then after that, you should now, this is A, you should be able to tell us what are the, you should be able to identify the types or components of the immune system that is actually talking about, that is affected. Oh, so this has to do with problem at CD4, and problem with the CD8, that is between cytotoxic T cell as well as T helper T cell. Okay, don't forget that this one is called T helper cell, and this one is the cytotoxic. So you should be able to know, identify the involvement of this in autoimmune diseases. You should be able to link autoimmune diseases to a kind of abnormalities in CD4 and CD8 pathway. In fact, when there is autoimmune diseases, that means the adaptive arm of the immune system is badly affected. It's badly affected. That is, something is wrong with that. Okay. Then, apart from that, you should be able to give out examples. This is very, very important. If you cannot mention examples and what happened, when we say example, you should be able to mention nothing less than 3. Be able to, and what are the clinical diagnosis of these examples that you are going to mention. Okay, so be able to tell us how can it be each of these. So after you must have listed them, to be able to tell us how it can be listed. I mean, it can be diagnosed. So, and how can it be controlled in case there is that. Okay, so that has to do with the fourth aspect on the third aspect, which is on autoimmune diseases. Okay, so the fourth aspect has to do with your understanding of classification. What are the types, classification, and functions of immunoglobulins. So the first one is that, what are they? Immunoglobulin, what are they? So answer this very big question. So then after that, you should be able to tell us, how do they work? How do they work? Whenever there is any infection, how are they being recruited? Then after that, so when you mention their classes, we have IG, that is, I told you, Gamder, that is IgGA, GG, IgGA, IgM, IgGD, IgGE, and in which of these where are they located? Location or localization, where are they located? Like if you have that of IgG is found in the blood, and this is the only IgG that can cross the placenta. This is the one that the parents can pass to their newborn. So you need to know that. Then A is found in the mucosa. A is found in the mucosa, respiratory tract, as well as gastrointestinal tract. So, and it is found to be abundant in this. Then IgM. So IgM, this is also found, and it is the first. It is the first IgG that will be recruited for primary immune response. Primary immune response. Okay. Then when we look at D, this is D. This D, it is not the function as well. The function is not well established. Although the function is not well established, but it performs its own role. Then we also look at the IgE, for example. IgD is found mainly in immature B cells. Okay, so then you look at that. After you must have looked at that, you now start to look at their functions. So, and basically, I want you to focus on 3 areas in their function. Number 1, you have to focus on their function in neutralization. Okay, how are they able to neutralize antigen? in case there is any infection. To be able to emphasize that. Then their role in opsonization. I told you the meaning of opsonization and what function do they perform in the complement system. So this is basically going to involve IgG as well as IgM. These two immunoglobulins are very vital, very vital, and because of their critical role that they play in complement system. So their role cannot be overemphasized. So that is how to go about the fourth one, which has to do with looking at the classification and functions of the immunoglobulin. So I said, what is the adaptive? What can you say about innate and the adaptive immune system? So this is very broad, and there is not going to be, you should expect that there must be, if not one or two questions on this that's going to demonstrate how do you understand, how do you understand, and what do you understand by immune system generally. That is the overview. Here we are expecting you to give us the overview, everything you know, that you have to make sure that you bring out very important points, important cogent points, overview of immune system. So you start with innate. What are they? We start with you continue with adaptive. What are they? What are their functions? How do they perform their role? And centrally, the first thing is that I want you to come up with that chart. On the chart, you should be able to tell us that we have innate, okay, then we have adaptive. When we have innate, what do they do? Which are the physical barrier, chemical and microbial, these are not these ones. You have to mention all of them. I need that chart. That chart is very important. Even if you know why the chart is so important. The chart, once you are able to understand the chart, or you have the chart, that is going to guide your discussion of when you want to make some important points under that. And apart from that, you'll be able to link the two together. How do they work hand in hand? You should be able to link them. Interaction, how do they interact? You should be able to talk about that. So you know it's going to be a very big question on that. Very big questions on that. Okay, so that is going to be the fifth one. Then the sixth one has to do with application of immunology in application of this course. That is application of your knowledge in immunology in laboratory techniques. So you find this very interesting, especially you have to link them with technique like ELISA, that is enzyme linked immunosorbent assay. How applicable is immunology in understanding that? So apart from that, you also need to talk about immunofluorescence. What do we understand by immunofluorescence? So I'm very sure you should be able to have them. Then you should also focus on Western blots. These are three important laboratory techniques that has immunological basis. That if your knowledge about immunology is shaky, honestly speaking, you might not be able to apply this. You might not be able to apply these techniques in laboratory. So in the diagnosis and treatment of some ailments, okay, especially when it comes to come with diagnosis in public health. So these are part of what it is expected that you should know. Okay. So now number seven is vaccination. What do we understand by vaccination and immunization? So what are the techniques? What are the basis of this? So you should be able to focus on active. What do we mean by active immunity and passive immunity? So please, you have to read more and focus more on this. Okay, you should be able to tell us what are vaccines, what do they do, history of vaccines. You should be able to give us very briefly the background, very briefly. Okay, then apart from that, you should be able to come up with what do we understand by what is called herd immunity. Herd immunity. So what do we mean when we talk about herd immunity? Should be your point of focus as well. Okay, so under that, I'm really expecting you to discuss active so passive immunity. What do we understand by it? Vaccine production or vaccine, the process of immunization, that is application of a vaccine in protection our population from infectious diseases. So please, you should be able to focus that. Then we're already getting into the end. So I want you to focus on the role played by Mary Mylon. I said this, Mary Mylon, in the transmission of typhoid fever. This woman, Mary Mylon, she was a cook in the former British Empire. She was a cook, and she was carrying typhoid fever, Salmonella typhi, as an asymptomatic carrier. And she was able to. They discovered that so many people from that population, they were already being diagnosed with typhoid fever, and the origin, the source of their transmission could only be traced to that woman or to that lady, and she was asymptomatic. So as long as she was still there, many people developed or contacted typhoid fever as a result of their contact, because she was the cook. She would only cook for them. So until she was now being exiled, she was sent on exile before they now discovered that the chain of transmission of typhoid fever was broken. And this one, you should be able to tell us more about some public health measures that could prevent typhoid fever, like food handling, proper food handling, improved sanitation, purification, sanitation, as much as you do. But I want you to emphasize, please read more about this woman called Mary Mylon, who was an asymptomatic carrier. And the last thing, the last aspect, which has to do with the ninth one, is what are the basic terms and terminologies in public health. We didn't have too much time. We didn't have too much time to focus on public health, but I want you to look at public health. You should be able to get yourself familiar with some of the terms and terminologies that we normally use in public health. This includes epidemiology. What do we understand by epidemiology? What is herd immunity? What is immunization? What do we understand by risk factors? What do we understand by incidence, prevalence, distribution, epidemic, pandemic, endemic. What do we understand by that? And later on, I want you to also focus on the role played by community education or enlightenment in the eradication of some of the diseases that are of public health importance. So we will also expect you to know all this. Now, lastly, let me now go to the grading system. The grading system is going to be a bit different from how you are going to be graded in the other one. Don't forget that your attendance is very, very important. I said attendance is very important. Then apart from that, you are given some kind of task, whereby a kind of presentation, group presentation, that I call it DIY. So your DIY here is very, very important. Although your DIY is going to also be included in other courses as well. But basically, in this, because you are expected to create awareness, with your do-it-yourself presentation. So that one will also help you in your part of what is going to be your grading system. So how it is going to be done, I think we are going to see that. Then summarily, just like I said, nobody is expecting you to write too long notes. Just in bullet form. Just highlights in bullet form, so that you don't need to be unnecessarily too detailed. But you should make sure that you demonstrate to us that you know what you are doing, so that at the end of the day, it's not going to be that you will just write notes without hitting the points. That is very, very important. Very, very important. And whenever you are going to give a chart or any illustration in form of diagram, you should be able to have a section in your response to those questions. You have a section that's going to annotate your drawings. That is going to explain, give us clues to what you have done so far. So just like when you will be expected to discuss classical pathway, alternative pathway, as well as a lectin pathway, you are expected to illustrate them in a kind of schematic drawing, so that you'll be able to tell us alternative pathway, classical, as well as lectin. What are those things that are involved? So this is going to be a kind of scheme, So I want you to not to forget that I said the first point of call should be component and mechanism of the complement system. After that, the basic principles, both biochemical, genetical, as well as immunological basis of ABO blood grouping system. After that, that of rhesus blood factors. So after that, you are supposed to know something about autoimmune diseases. What are they? Their definitions and examples and their diagnosis. After that, we also expect you to be able to talk more. You should be able to tell us more about the classes or classification and function of immunoglobulins. Apart from that, you should be able to tell us more about the components, general overview of immune system, both innate as well as adaptive. And you should be able to tell us the components, the mechanism, and how do they intricately work together in form of a network of proteins or network of activities that has to do. Then application of immunology in laboratory techniques with a different focus on ELISA, that is enzyme linked immunosorbent assay, immunofluorescent, and as well as Western blotting. You should be able to know that. After that, we also expect you to know more about immunization. Focus on immunization and vaccination. What do you have to say? You don't necessarily need to be too detailed, just like I told you. Just let us know. Then the role played by Myrimylon in transmission of a typhoid fever. So then you are going to cap it all with basic terms and terminologies that you are supposed to know in public health. So having done all this, I'm very sure that you are going to have a very successful outing in immunology and public health. So thank you for your attention.